Dr. Lyn Patrick here. I want to give a brief introduction to Bob Miller's webinar because I think this is so critical that we understand iron physiology and iron pathology. And I want to start out by introducing an article that I reviewed in a recent podcast. This is research that's done by Dr. Lilian Calderon-Garciduenas at the University of Missoula in Montana. She also works in Mexico City with several medical schools there. And what she has found, and a lot of this information, interestingly, was done through autopsy, is that there are nanoparticles, many of them iron, that are found in human hearts, both in infants, in children, and in adolescents. And these iron and lead, also nanoparticles, from air pollution, from friction, what are called combustion and friction-derived magnetic air pollution, nanoparticles, meaning both friction from brakes and combustion from gasoline, that these nanoparticles caused systemic oxidative stress, were actually found in the circulation systemically, induced endothelial dysfunction, induced autonomic nervous system dysfunction, induced arrhythmias, and she also found these same metal nanoparticles in the brains of both children and adolescents in Mexico City, correlating with, I'm going to show you this, neurofibrillary tangles and ApoE4 carriers. So, very, very compelling information. I'm going to try to attach these articles. In fact, I will upload them in the blog area so that all of you can see the actual research. I think it is so incredible. But the bottom line here is that these magnetized nanoparticles, so it's not just iron, it's magnetic iron, these nanoparticles that were the same nanoparticles, both iron and lead, that were found in the urban atmosphere, were in such prolific abundance. They were in such high abundance. And again, Mexico City, most polluted city on the planet still, that she determined that it really is a serious public health concern. Another thing she did mention in this article that I don't have a slide for, but you'll read it, is she said, given that these are magnetized nanoparticles, certainly their pathology, their damage that they cause, would be accelerated when exposed to electromagnetic radiation or electromagnetic frequency. So very important information. This was just published in 2019 and reviewed. The other one that I want to show you, the other article I want to show you, just so you'll be primed for Bob's talk, which is so important, is about iron metabolism in the body. So this is just an article about iron metabolism and the liver, and basically talks about how the liver is really metabolism central for iron physiology. The liver actually does sense both iron stores in the liver, but also iron stores systemically, and from that control center actually regulates the hormone hepcidin. Hepcidin is a hormone produced in the liver by the hepatocytes, and it increases in response to transferrin saturation or inflammation, inflammatory cytokines, which we know are produced by environmental pollution as well as infection, right? The receptor for this crucial hormone is ferroportin, and Bob's going to talk about ferroportin, the only known iron exporter in mammals. And of course, it's directly related to the efflux of iron from the liver as well. And so it gets downregulated by inflammation. So it's a kind of the opposite of what we think of as an acute phase protein. So ferritin is an acute phase protein. And in inflammation or infection, ferritin levels go sky high. We also know they're elevated in non-alcoholic fatty liver disease. as they are in other liver problems. So intracellular iron trafficking is really controlled by this system of hepcidin and ferroportin, and Bob is going to talk about that. So I'll put both these articles up in the blog. Enjoy, and just know that this is a completely different way of looking at iron trafficking in the body. And we know that iron is one of the main causes of reactive oxidant stress in the human body. We don't like to think about that because iron is a necessary nutrient without which we can't make heme. Without heme, we can't build bones. We can't make cytochrome P450 detoxification enzymes. We can't do a variety of things that necessitate iron. But it is really the source of iron, the level of exposure, and the ability of the body to get rid of it, which is what Bob's going to talk about. Especially for those of us, and Bob will mention this, I'm sure, who have lineages either from the British Isles or are Ashkenazi Jews. And so for yourself or your patients who have that genetic background, that's really important to pay attention to. All right, so enjoy.
Hi, Dr. Patrick. Once again, an honor and a privilege to be presenting that we can get some information to your practitioners as to how they can support detoxification. This is the third webinar that we're doing. And the first one, we talked about the importance of NADPH and how it's involved in recycling so many things and having so many functions. So if you haven't listened to webinar number 1, certainly encourage you to do so. Then in webinar number 2, we talked about how the NOx enzyme is taking that NADPH and using it potentially under some circumstances excessively to create mast cells and histamine and create lots of reactive oxygen species. Now, in that webinar, we talked about how iron is one of the stimulators of that NOx enzyme. So our webinar today is how dysregulation of iron is contributing to inflammation and poor detoxification of environmental toxins. So obviously, I don't need to tell you folks that iron is critical for many functions. However, excess or unregulated iron can be extremely inflammatory and cause damage and impair detoxification ability. This is called the Fenton reaction. When iron combines with hydrogen peroxide, hydroxyl radicals are made. Now, the uncontrolled action of hydroxyl radicals, considered the most damaging free radical by far, can have devastating effects within the body. It's a third-generation species of radical which is derived from hydrogen peroxide, which in turn is derived from the superoxide radical through the action of the enzyme superoxide dismutase. Quick side note, early on in our consulting work, we would think that superoxide is bad, superoxide dismutase is good, therefore giving it to people will be helpful. Most of the time it is. But for some people, they had to stop because it made them worse. What a paradox that was. And I'm going to be showing you in a moment why that can actually happen. But back to hydroxyl radicals. The dangers of it has been highlighted by Dr. Ryder. If the function of radicals is to destroy molecules and tissues, then the hydroxyl radical would be the radical's radical. It reacts at diffusion rates with virtually any molecule found in its path, including macromolecules such as DNA, membrane lipids, proteins, and carbohydrates. It can induce strand breaks as well as chemical changes. Damaged proteins, many of them crucial enzymes and neurons, lose their efficiency and cellular function wanes. Protein oxidation, including the brain, has been associated as one of the functional deficits associated with aging. So that's why we spend a lot of time researching hydroxyl radicals and how to make less of them. I probably show this chart 5 times a day. So here's what happens. Inside our cell, as we make energy, sometimes an electron flies off, combines with oxygen, makes superoxide. The body in its wisdom says, I need some superoxide dismutase to make hydrogen peroxide. Then we need catalase glutathione peroxidase, thioredoxin, to turn that into water. If that doesn't happen, or doesn't happen as efficiently as it should, combines with iron in Fenton reaction to make hydroxyl radicals, damages proteins, carbohydrates, and DNA, can oxidize your fats. Will also combine with inos and superoxide to make peroxynitrite, another very oxidizing agent that suppresses the immune system. So what are the risk factors for this? Genetic factors, or even epigenetic. Somebody eats too much iron, low ceruloplasmin. We all know ceruloplasmin is what carries it around. Ceruloplasmin is copper dependent. We need vitamin A. Or if you would have, low catalase, anything that's happening to your glutathione or thioredoxin. And back to one of my favorite subjects, NADPH. Please listen to webinar number 1 and number 2 to learn about NADPH. It takes your oxidized glutathione, turns it back to reduced. Your oxidized thioredoxin turns it back to reduced. Not enough NADPH, this doesn't work as effectively as it should. Fenton reaction occurs. In the first study that I did on Lyme disease that won the research award in Helsinki, Finland in 2016, we showed that those with chronic Lyme disease had 5 times more likelihood of having the genes that would cause them to overabsorb iron. So again, iron critical, excess or not controlled can create all kinds of havoc. Now, this is just a lot of data on here that you just may want to study. But what I did is I listed the genetic mutations that could impact your solute carriers, which carries your copper for ceruloplasmin, your SOD1, which is a copper-dependent superoxide dismutase, copper transporters, ceruloplasmin. So what we do when we see a lot here, you want to measure your red blood cell copper. If you have a lot of mutations here, measure your ceruloplasmin, because one of my favorite sayings is genetics mutations is a potential. So we see people with mutations in ceruloplasmin, we measure, ceruloplasmin is fine. Other people, and there's probably other factors, the ceruloplasmin is low. But if you have mutations in your copper transporters, and then you've got mutations in your ceruloplasmin, that combination often leads to low ceruloplasmin, and then you're more potential for the iron to become an oxidizing agent. I'm sure you all know that beta carotene, things you get in your carrots, needs to be turned into vitamin A. Mutations in the BCMO1 can inhibit your beta carotene to vitamin A. So what do you do if you see this? Measure your beta carotene and vitamin A, see what's going on. To me, this is one of the more clinically significant and not easy to detect, because ferroportin is your only iron exporter, your SLC40A1. If we don't export it, the iron can get stuck inside the cell and cause all kinds of damage. One of the things that we've observed over the years, many times people have genetic issues where they overabsorb iron. They have the ferroportin enzymes, and blood work comes back anemic. And when I tell the folks, oh, it looks like you overabsorb iron, they say, oh, no, that can't be true because I've been told I'm anemic my whole life. Well, if that excess iron is being chewed up to make hydroxyl radicals, blood levels can be low. So I'm sure you've all experienced this. If someone has, low hemoglobin or low iron, and you give them iron and they feel worse. Now, we have many times seen people with low iron. We support their catalase, their glutathione, their NADPH. Don't give them iron, and their iron levels rise because we've slowed down this process here. So is there times that iron supplementation is needed? Of course. Is there times where iron supplementation can be the worst thing you've done? Yes. So I think that's why we have to be very aware when's the right time to give iron. Because if the iron is low because Fenton reaction is occurring, you can be pouring gas on the fire by giving iron at the wrong time. All right, now how do we see what the iron's doing? Well, there's iron transporters, there's iron absorption. Heme needs to be broken down by the HMOX enzyme and put into ferritin. So if someone has a lot of mutations on HMOX, their ferritin might be low, and it's not that they need more iron, they need support of the HMOX enzyme. Magnesium transporters. Magnesium, critical for inside the cell, and there is some belief that excess iron banging around can actually inhibit in some ways the magnesium. So in our genetic reports, we look at all of these. And again, I often say this is the 3D chess game played underwater. If somebody says, you know, this SNP does that, I think we're missing the boat. We've got to look at a huge picture. We've also got to look at the epigenetic factors. Okay. Now, we talked about in our earlier webinars about the importance of NADPH. So I'm not going to repeat these. We cover this in the first segment, but mutations here can limit the production of the NADPH. Webinar number 2, we talked about how activation of the NOx enzyme or overactivation of it can impede this. Glutathione peroxidase, catalase, PRDX, and thiodoxin, and recycling of thiodoxin are all involved with clearing the hydrogen peroxide. So serious mutations in any of these. I just did a consult with a physician. I oftentimes, help physicians look at the genome to get clues. One person was incredibly ill, and their genome looked much, much better than everybody else until we got to the recycling of thiodoxin. It was a train wreck. So that may have been one of the issues that caused the struggles that they were having. Now, we all know that copper is a mineral that's found throughout the body, helps the body make red blood cells, forms collagen, antioxidant, of course, too much copper toxicity. And we don't need much. But if we don't have enough to make our ceruloplasm, we're going to be in trouble. So these are the enzymes that are involved in the proper use. They're the solute carriers. What's interesting, many times people of Irish descent have mutations here. Just a Bob Miller wild idea. I have to wonder if people who have red coppery hair, maybe not carrying the copper properly and it's being excreted. It'd be fun to look at people with red hair and see if they have more copper, but just observing, that's all. No peer-reviewed studies, nothing else. Just observing. Many times people with coppery colored hair seem to have a lot of mutations in these copper transporters. Might be a coincidence. SOD1 provides instructions for making the SOD, which is copper dependent. So once again, you could have no mutations in SOD1, but if you don't have any copper to feed it, you're not going to make it. Here's the study. Expression of mutations in SOD1 can lead to increased generation of hydroxyl radical-like activity. Sodded copper. SOD1 binds copper and zinc in one of the 3 superoxide dismutases responsible. Keep in mind, superoxide is what stimulates the mast cells. And I'm sure all of you who are, doing this work have noticed just a tremendous increase in mast cell activation. Is this the only cause? Of course not. Do I believe this is a factor? Yes. ATOX1, ATP7A, ATP7B. The ATOX plays a key role in copper homeostasis as it delivers copper from the cytosol to transporters. ATP7A is a copper transporter. ATP7B, also another copper transporter that's involved in Wilson's disease. And again, these are serious genetic mutations. And are they being expressed? We don't know. But this is where you then do the testing. Look at the serum copper. Look at the red blood cell copper. Look at the ceruloplasm. I always tell practitioners, genetics is not a diagnosis. It's not a for sure. Think of it waving at you. Say, consider looking over here in your assessments. And many times it tells you to look places that you never would have thought about. So here we go. If many variants, consider measuring RBC copper, measure ceruloplasm. supplement gently with copper if needed. Okay, so you all know ceruloplasmin protein binds most of the copper in plasma and is involved in the peroxidation of Fe2 to Fe3. Variations cause, it's a hard word to say, causes iron accumulation and tissue damage and is associated with diabetes. Vitamin A is also needed to make ceruloplasmin. So keep an eye on that ceruloplasmin. BCM01, the conversion of beta carotene to vitamin A. Now, have you ever seen anyone who's trying to do all they can for their health, and they drink a lot of carrot juice and they turn orange? I'm fairly certain they've got some trouble converting their beta carotene to vitamin A. So this is where you need to look at other sources of vitamin A. One good source, of course, is cod liver oil. So what do? If there's a lot of mutations, consider measuring beta carotene and vitamin A and measure your ceruloplasmin. We don't do a lot of it, but when we see a lot of mutations in BCM01, we tell folks, see your primary care physician, get them to measure beta carotene and vitamin A. For some people, it's the issue. Their beta carotene is high, their vitamin A is low. Other people, it's normal. Again, there's probably so many factors. So that's why that old adage of you don't treat the SNP, but consider this waving at you to look at something. So look at this study, high iron levels in Alzheimer's. It can lead to the production of free radicals that damage neurons in your brain. It is believed that iron accumulates at high levels and is extremely reactive in the plaques found in the brains of Alzheimer's. It collects in the brain regions associated with memory and thought processes, which are gradually lost as Alzheimer progresses. So research has shown that reducing excess iron in your brain can alleviate Alzheimer's-like symptoms. We've seen that. People that get the iron under control, their memory starts coming back. Non-alcoholic fatty liver disease. 1/3 of the patients with non-alcoholic fatty liver disease show signs of disturbed iron homeostasis. All the references are down at the bottom. It's small print, but there's where you can go to read the study. Now, this is interesting. The literature is showing NADPH oxidase uses iron to create the superoxide. If this would be overactive with the NADPH steel, there's the potential for excess reactive oxygen species, and then again, less NADPH for its critical functions. If you didn't see webinar number 2, highly encourage you to watch that. So here's a study. Iron accentuated reactive oxygen species released by NADPH oxidase in activated microglia and contributing to oxidative stress. So iron significantly increased the reactive oxygen species production and neurotoxicity, suggesting that the iron contributes to this oxidative stress and may be a valuable therapeutic target for injury or neurodegeneration, suggesting that NOx2 and NOx4 might be ones to look at in that issue. SLC40A1, one of my favorites, makes ferroportin the only iron exporter. It transports iron from the small intestine into the bloodstream, plays a role in the metabolism of iron. Different variants in the SLC40A1 gene affect the ferroportin protein in different ways. Just us observing in our research. When people have a lot of mutations in SLC40A1, these individuals have a lot of inflammation that nobody can seem to figure out why. Is it the only cause? Probably not. Is it a contributing factor? I highly believe so. So this one transports heme into the intestinal cells. Whenever you see SLC, that's solute carrier. This is deviant metals involved in iron absorption. These are involved with microcytic anemia with iron overload. Of course, the one that's very well known is the HFE. HFE H63D represents a SNP that accounts for a mild form of hereditary hemochromatosis. This is the one that was 5 times higher in those with chronic Lyme disease. So when they're homozygous, this is where we have hemochromatosis. but even heterozygous can cause a slight increase in the absorption of iron. So if these people then have ferroportin issues, if they might have low ceruloplasmin, low vitamin A, cook with iron skillets, it's those multiple factors that may cause the iron to be creating through the Fenton reaction, the hydroxyl radicals. Ferritin light chain provides instructions for making the ferritin light chain the major intracellular iron storage protein. I had a optometrist tell me that he finds this incredibly correlated to cataracts. Quite fascinating because it's related to the ferritin in the eye. ACO1, variations in this gene have been associated with hemochromatosis. So what we do in our software, Functional Genomic Analysis, we have a whole panel that shows you all of these. And you have to look at them together, which takes me back to, saying this SNP creates that. May be true, but we have to look way beyond that. We have to be thinking globally. Do I have multiple factors that builds upon itself? TRF2 mediates cellular uptake of transferrin-bound iron. And again, variations in this gene have been associated with hereditary hemochromatosis. Could you have mutations in TRF2 and iron be doing quite well? Absolutely. But what happens when you've got problems with your copper, your vitamin A, your ceruloplasmin? You might store it inside the cell, and then you have this. In my opinion, we need to do more research, but this is when this becomes significant. Transferrin factor. Okay, variations in this gene have been associated with iron deficiency and iron metabolism disease. Now, we're big fans of the heme oxygenase genes. Why? Because here's where your own heme uses our good buddy NADPH and cytochrome P450 to put your iron into ferritin, to make your biliverdin so that it can go into bilirubin, a very powerful antioxidant molecule. And this CO, we don't have it on here, but it goes on to stimulate NRF2, KEAP1, DNA repair enzymes. So when you've got mutations in HMOX1 or HMOX2, there's a very strong chance that this heme is not going. You can see your iron in the middle. So what happens to the iron? Potentially it becomes dumped and becomes hydroxyl radical. Heme oxygenase is an essential enzyme in heme catabolism. But again, it forms the biliverdin. The biliverdin can turn into bilirubin and carbon monoxide. And again, it's that carbon monoxide that stimulates the, who'd ever thought the carbon monoxide could have benefit? But of course, a lot of it kills you, but a little bit of it stimulates the NRF2. Bilirubin is a compound that breaks down heme in vertebrates. The activity is highest in the spleen and has an association with pulmonary disease. So here again, we want to go back to this chart showing that NADPH, critical. If you don't recycle your oxidized glutathione, if you don't recycle your thioredoxin, and also if you don't have this to work on putting your heme into storage, you're setting yourself up for this process of Fenton reaction and hydroxyl radicals. So again, 3D chess game, NADPH, production of glutathione, production of thioredoxin, production of catalase, perhaps overproduction of superoxide, too much iron or unregulated iron, multiple factors. But again, this goes back to one of my favorites, hydrogen water, because it won't, slow this process down a little bit, but OH minus plus H2 equals H2O. So NADPH, and I'm not going to talk about that a lot. We did in the previous webinars. To clear the hydrogen peroxide, you need glutathione peroxidase and thioredoxin. Again, this was covered in the NADPH webinars. Go back and listen to those if you're just listening to this one. Glutathione peroxidase is what uses glutathione to clear the hydrogen peroxide. Our online certification course covers all of that. Catalase supports the clearing of hydrogen peroxide. So if you remember earlier, I said I was a little surprised when taking superoxide dismutase made people worse. What we found is you got to get some catalase first, or support NADPH, or support glutathione peroxidase before you gave SOD. Okay. Peroxidation encodes the members of the peroxidin family of antioxidants enzymes, which reduces hydrogen peroxides. Now, let's go back to hydrogen peroxide. There are people who, I think there was a book one time, The 1 Minute Miracle Cure, which was taking hydrogen peroxide. I would imagine the theory behind it, hydrogen peroxide kills pathogens. So again, hydrogen peroxide is not bad. But when it's excess or not cleared and combines with iron, we can create a problem. So how many people read that book, started taking, food-grade hydrogen peroxide, and rather than getting better, got worse? And again, some people do treatment with hydrogen peroxide because it kills pathogens. Is that a good therapy? Yes, unless you've got this iron turning it into free radicals, then it can work against you. So this is why I believe we need to go back to personalized care. What can be right for one person can be wrong for the next. Together with thiodoxin and NADPH, this system's general description is as a method of forming reduced disulfuride bonds in cells. And again, electrons are taken from NADPH as a defense against this oxidative damage. Please watch the webinar that I did on NADPH and NADPH oxidase. I believe there's strong evidence this is being overused by environmental toxins in all of us, amplified in those who might have genetic mutations that either don't make enough or something's upregulating the NOx enzyme. Thiodoxin reductase, the selenium protein whose expression and function depends on an adequate supply of the trace element selenium and recycles your PRDX. I mentioned earlier, I was doing helping out a functional medicine doctor because he said, I can't figure this person out. They were really doing great until we got to here. Massive amounts of homozygous variants. And you would have never known that without looking at the genome. So together with thiodoxin and NADPH, this system's most general description is a method of forming reduced disulfide bonds. We talked about that. That's why I'm a huge fan of hydrogen and functional genomic and nutrition. Just private label this, but I'm sure you're all familiar with this. It's the pill you drop in the water, knocks the hydrogen loose, does quite a bit. Evidence-based health benefits, converts the body's most harmful free radicals to water, regulates the body's antioxidant system, including SOD and glutathione, supports cell metabolism, proven to defend your body against free radical damage. I think I said this in another webinar. If I was limited to one thing and one thing only, I think it would be hydrogen water. Now, what we did at functionalgenomicnutrition.com, we formulated, I believe, the most comprehensive way to support the proper use of iron. For those who overabsorb, it slows it down, supports hydrogen peroxide clearing with catalase and glutathione peroxidase support, antioxidant support for inflammation. So I'm not going to go through all the ingredients, but vitamin A to support the efficient use of iron, copper to support production of adequate ceruloplasm, clove, one of the strongest antioxidants, but slows the absorption of iron, rhodiola, vitamin E, and catalase to support hydrogen peroxide to water conversion, riboflavin and selenium to support the function of glutathione peroxidase to turn hydrogen peroxide into water. Now, clearly, this is a great formula. However, some people might say, oh, this person might be a little high in copper, maybe I shouldn't do that. Man, they've got a huge histamine problem, a little concerned about all that clove. That's why we worked with personalized nutrients that actually in the software, you can go in, pull this formula up, take anything out that you want, decrease, increase, you whatever you want. So for example, if you test and see that the vitamin A is really low, you might want to jack up the vitamin A palmitate. If the vitamin A is good, you might want to bring it down. If the person has a boatload of COMT, you may want to take out the quercetin or decrease it. Okay. And if the person has a huge problem with histamine, I mean, if the histamine is just over the top, you may want to decrease or eliminate the clove. So we'll be doing training on that as to how you as a practitioner can make a personalized product for the individual. The other advantage is, say for example, you want to give a couple of these, you can combine them all together into one capsule. Personalized nutrients makes it, sends it to them. Unprecedented ability, but I believe in these times that we're living today, we need that unprecedented capability. We'll be teaching you in webinars how you can actually formulate, take what I've done here, tweak it. So here's the ingredients: quercetin, skullcap, luteolin. We talked about luteolin to suppress the Fenton reaction. Alpha-lipoic acid and acetyl-carnitine to support oxidative stress. Olive leaf for the inflammation. So here's how we can help you. The Nutrigenetic Research Institute, that's all we do, research, but we have online education, live conferences. Go to our website, put that in the link. Functional Genomic Analysis is the software that analyzes this, organizes, interprets the SNPs. Your genomic resource is the DNA test kit, the supplements, functional genomic nutrition. We've also worked with professional health products. And then again, real exciting, you as the doctor can make your own supplement based for that individual. So here's the website, nutrigeneticresearch.org. This is where we have the links to our education online classes. Here's the website for the online certification course. The first 3 modules are free. And this is just the test kit, looking at 200,000 SNPs. As a practitioner, you order it, send it to your patient, they send it back or give it in the office. This is not available to the public. This is not like some of the things that are available online. Doctors only. And then this is the kit that is sent to them, the Your Genomic Resource. We partnered with Thermo Fisher and Brooks at Rutgers. Of course, you need the software. Unbelievable price, $199. $235 for the buccal swab for those who can't get the saliva, includes all the reports. And there's the website, functionalgenomicanalysis.com. If you're a health professional, you can request an account for free. You get to play around with it. If you have an old 23andMe, version 3 or 4, play around with your heart's content. Then it's only $25 a month. And there's the contact information for Yvonne Lucchese, Executive Director. Her phone number is 717-466-5700. Yvonne L at DNAsupplementation.com. So let's summarize. Iron, of course, critical. Gotta have it. Can't live without it. However, in excess or not carried around properly, one of the nastiest free radicals. Additionally, it can feed the NOx enzyme to stimulate the mast cells, that stimulates the histamine, that wears out your adrenals. Since we need progesterone to make your cortisol, could be impacting your hormones, could be creating all kinds of histamine responses. Just as important, or possibly more important, is using up that critical NADPH, which is needed to recycle your antioxidants. So excess iron, unregulated iron, I believe is one of the most important things to look at. And that's why in our pyramid approach, it's the first thing we analyze. We look at the copper, the vitamin A, the ceruloplasmin, all the things that cause overabsorption of iron, and then all the things that support the clearing of hydrogen peroxide. That's the first step I believe you need to take in a functional nutrition approach. Make sure that's not happening. Just sneak preview, the second step is looking at NOS uncoupling, then glutamate, then heme cycle dysregulation. And by the way, the heme cycle is so important because we need the heme for all your detoxification, and lead inhibits the ALAD enzyme, which can mess up your heme cycle and can totally disrupt your ability to detox. And also those porphyrins will then block the GABA receptor sites. So we could do a whole other webinar on just how lead just can impact that heme cycle. So if you are intrigued by this and you're willing to work hard, this is for you. If you're looking for a one-page summary that says what to do, don't bother. Because sometimes we've had doctors who have done that. It's like, well, where's the one page that tells me what to do? I don't have time to study this. Then fine, then don't. So I just want to give you a heads up. I don't want to waste your time if you're not serious. But if you are intrigued and being a detective, digging deep, looking, I believe this is a tool that could be very helpful for you. So Lyn, thank you for this opportunity. What an honor and privilege to be able to present this information. And we're looking forward to meeting everyone whenever EHS is. At the time of this recording, there was question whether or not it would have to be postponed. But whenever it is, we'll be there to answer your questions. Or in the meantime, feel free to reach out to us or take the online certification course. If the event doesn't occur and you already blocked that time out, that might be a good time to go through the online certification. So, Lyn, again, thank you for this opportunity, and any closing comments by you?
Thank you, Bob. I have a lot to say, but I think that I will have summarized it at the beginning of this recording. So thank you so much. This is such incredible information, so crucial to those of us who are practicing environmental medicine. I'm really grateful. We'll look forward to more from you. Absolutely. Thank you so much.
My pleasure. Well, that was fun.